So we're moving from plants to fungi, and in situ expression of carbohydrate active enzymes from Annika Mosier.
Okay, thank you. I want to thank the organizers for inviting me to present my work here today. I'm going to be talking about carbohydrate active enzymes in the acid mine drainage system. And I want to point out that this work is part of a broader project involving a number of people who are listed here at the bottom of the screen. We heard a nice talk this morning about algal biofuels, and another important area of research as an alternative to alternative energy to fossil fuels is that of the second generation biofuels from lignocellulosic plant materials. There's still significant technological bottlenecks in this process in the efficient conversion of biomass into biofuels. The enzymes currently used in the process can't meet the growing demand for economically viable biofuels due to their low activity, their high cost, and their poor stability under the required operating conditions. Therefore, it's essential that we continue development and search for novel enzymes that can be used in this process and mitigate some of those bottlenecks. Carbohydrate active enzymes are a broad class of enzymes that build and break down complex carbohydrates, and they're involved in several steps in the biofuel production process, including the breakdown of plant cell walls. Microorganisms, including some bacteria and fungi, are very efficient decomposers of biomass and often have many CAZymes, or these carbohydrate active enzymes, and therefore they're a prime target for novel enzyme discovery. One strategy for identifying novel enzymes in these microorganisms is by mining their microbial genomes. We're using acid mine drainage, or AMD as it's called, as a model system to systematically examine carbon cycling in microbial communities at a molecular level. In this particular work that I'm going to talk about today, we're looking at the expression of these CAZymes, the carbohydrate active enzymes, specifically by eukaryotic fungi within the AMD biofilms. The AMD solution is characterized by low pH. We see pHs around 1 usually, as well as high temperatures ranging from 40 to 50 degrees Celsius. We also get high metal concentrations. Therefore, we expect that if we see carbohydrate active enzymes expressed by these organisms in the AMD solution, and those CAZymes are then excreted from the cell, that these enzymes have adapted to those extreme conditions, and therefore they may prove useful in the production process of biofuels because they likely have higher activity at these high temperatures and low pHs, which are conditions very useful in the production process. This map here, or figure here, is showing the typical biofilms that we find in the AMD solution. In the early stage, developmental stage biofilms, we get colonization by pretty much one major type of bacteria, the leptospirillum, which oxidize iron and fix carbon dioxide. As the biofilm grows and develops, it thickens and it also diversifies. And we see other bacterial species present, as well as some archaea and eukaryotes. The pictures shown in the top panel here in images A and B are those classic floating biofilms that we see in the AMD biofilm. You can see in the light pink on the fringes of the AMD solution are these early developmental stage biofilms that have predominantly one main bacterial species. The dark pink are these later developmental stage biofilms here over on the closer to the stream shore in connection with the pyrite minerals. The microscopy image is showing the eukaryotes in these late developmental stage biofilms, and you can see them in green and orange. I should point out that the total diversity of the system is very low. So in these classic floating biofilms that form at the air surface interface, we see somewhere on the order of 20 species. So we're dealing with a really simple system in essence. There are certainly a lot of complexities to it as well, but from a genomic perspective, it makes it a bit easier to do some of this work. The bottom images here are showing a different type of biofilm that we see in the mine, and we call it a fungal streamer biofilm. You can see in the microscopy images that we get these really thick bundles of filamentous fungi in these fungal streamer biofilms. If we look at the diversity of the eukaryotes in these two types of biofilms, we see different situations. In the classic floating biofilms that have been really well studied. The pie chart at the top here is showing that there's a diversity, some diversity of eukaryotes belonging to both the protists and fungi. In these floating streamer biofilms on the bottom, this phylogenetic tree shows a purple arrow pointing to Actinomycetes richmondensis, and it turns out this is the only eukaryotic species that we see in these fungal streamer biofilms. Actinomycetes richmondensis belongs to the Dithydomycetes in the Ascomycota phylum of fungi. So then we wanted to look at the CAZyme expression, again, these carbohydrate active enzymes in the AMD fungal streamer biofilms. And as I mentioned, this is dominated by one species of eukaryotes, the A. richmondensis fungi. To target the CAZyme expression by these organisms, we used metagenomics to look at the bulk DNA from the biofilm community, metatranscriptomics, and I should put meta in parentheses here because what we did is we pulled out the eukaryotic mRNA in particular, and since we only have one species, it's sort of a fake metatranscriptome. But we took a metatranscriptomic approach. We also did a metaproteome of these fungal streamer biofilms to look at the proteins. It turns out that we're also able to isolate Actinomycetes richmondensis in the lab, and this work was done by Steve Singer. And therefore, we were able to get a genome directly from this isolate and use that to assist our metagenomic assembly of A. richmondensis. This table here is summarizing the genome assemblies for both the isolate and the metagenome. And the top 4 or so rows just give you an overall sense of the assembly of the genome from A. richmondensis. We use the isolate genome to assist the metagenome assembly, so you can see that the stats for the metagenomic genome assembly of A. richmondensis is slightly better than that of the isolate. In both cases, we see that the genome size is on order of 26 to 29 million base pairs, and we get about 10,000 to 11,000 gene models. We then looked at the transcriptomics of A. richmondensis in the fungal streamer biofilm, and from 24 gigabases of data, we see 10,149 transcripts. If you remember what I just said, we only see about 10,300 up to maybe 11,000 genes predicted in the genome. So we're seeing a transcript for almost every gene. So certainly this may be due to the high sequence coverage, but it's also possible that it's not a true biological signal, that there's some error there. This figure on the bottom is simply just to show the high dynamic range of transcript abundance that we see. We then looked at the metaproteome from the fungal streamer biofilm. We took our sample and we did 2 replicate runs on different mass spectrometry instruments. And in total, we found about 4,700 unique proteins from the AMD fungal streamer biofilm. Of those, about 1,100 were specific to the fungi, the A. richmondensis. This figure on the bottom here is showing a bar chart distribution of the taxonomy of the different types of proteins that we see. And in the bottom, I've highlighted Actinomycetes richmondensis, and we see that about 38% of the proteins were related to this fungi. So then to get at the carbohydrate active enzymes in particular, we went back to the genome and we annotated CAZymes using the CAZymes analysis toolkit, which is an online portal developed at Oak Ridge. And in this system, or in this portal, we did annotation based on PFAM similarity, where we set an e-value cutoff of 10 to the minus 4. And we also did a sequence similarity search with known CAZymes and set an e-value cutoff of 10 to the minus 6. In these sequence similarity searches, we also set a consistent length and domain stringency. So it's important to note that these are putative CAZymes here, that we certainly haven't tested the activity of these enzymes, but it does give us a pretty good sense of the overall picture of the CAZyme composition of the A. richmondensis genome. The different CAZyme classes that we see are listed here in the table and the general functions for each of those classes. We can see about 185 glycoside hydrolases, and of those, there's potentially 31 putative cellulases that are acting on the hydrolysis of cellulose. Among the other families, we see the carbohydrate binding modules, the carbohydrate esterases, glycosyl transferases, and the polysaccharide lyases. we've heard about a lot of these over the course of the meeting. It turns out that the number of GHs, the glycoside hydrolases, and the GTs, the cholesteryltransferases, there tends to be a general correlation in the number in the genome relative to the number of genes in that genome. This is the same is true here for the fungi. This plot is from Berkeley et al. In 2011, where they looked at the number of GHs and GTs for 10 different fungi relative to the number of predicted gene models. I've superimposed our data here from A. richmondensis, and that's shown in purple. You can see at the top that the A. richmondensis genome has approximately a similar number of GHs relative to the number of genes in the genome, similar to that of the other fungi. For the GTs, however, we see that A. richmondensis has fewer GTs relative to the number of genes, and we're still unsure, we're still examining this. It may be differences in the annotation of these GTs, or it may also be that A. richmondensis is certainly a unique fungi growing in a strange system with extreme conditions. So it could be that the comparison is just is irrelevant to these other fungi. So this plot is showing the expression of these CAZymes in the environment, in these fungal streamer biofilms, based on the transcriptomics and the proteomics. So in green are bars showing the number of genes for each of these CAZyme classes. And in light blue, I've shown the number of transcripts. And as I mentioned previously, we see essentially every gene is transcribed. So the same is true here for the CAZymes. I arbitrarily called abundant transcripts as those that are in the top 50% of the transcripts in terms of abundance, and those are shown in the dark blue bars. In yellow, you'll see the number of proteins that we saw in the proteome from the sample, from the fungal streamer biofilm. And although the numbers look significantly lower than the transcripts and certainly a lot lower than the genes, we are still excited to see that a lot of these CAZymes are present in the proteome, because remember that this is coming from the bulk community. So the number of proteins that we get back from the fungi in that community is only a certain percentage. And then to see these CAZyme proteins related to the fungi suggests that they're highly expressed in the environment. This table is showing the top 10 most abundant transcripts from the whole transcriptome of the A. richmondensis fungal streamer biofilm. And you can see that I've highlighted in purple that 3 of these top 10 most abundant transcripts are GHs, which is nice to see. You can also tell from the other transcripts that it appears that carbohydrate metabolism is a highly transcribed function by these fungi. We looked at the data in a slightly different way. Again, this is transcriptomic data. By narrowing our search to the GH families in particular. We did a summed expression rank as well as an average expression rate rank for each of these GH families that we find. And these were the most, consistently the most highly expressed GH families. You can see I've identified just the general function of these, each of these GH families. And there tends to be, we see breakdown of trehalose and cellulose, as well as glucans and galactans. We can also then go in and look at the signal P of each of these predicted genes and get a sense for genes that are putatively, or excuse me, proteins that are putatively exported from the cell. If we look at the proteome and look at the most abundant proteins in the GH families, we see the same story. We see the same GHs coming up, 13, 16, 72. However, I've highlighted in purple, we also see GH10 coming up, which is a family of xylanases. And so it's suggesting that these are among the most abundant proteins in the fungal streamer biofilm. So then we can put this information in the context of the environment where these fungi are living. And Zhao et al. In 2010 looked at the composition of the EPS of these biofilms and identified the most abundant sugars. And that's shown here in this table. And we see some of those sugars that we just identified in the fungi CAZymes. We see galactose, we see mannose and xylose. So it's neat to see that some of the most abundant sugars in the EPS are then acted upon by the fungi in the system. We can also use the genomic data that we have from the other community members in the biofilm and see who's making these sugars. For instance, we can see that some of the archaea are making, well, have the genes to make trehalose and mannose, and we see that, that the leptospirillum, the dominant bacteria, likely produces cellulase. So together, we're starting to get a bigger picture of who's making the sugars in the biofilm, the composition of the EPS in the biofilm, and then the fungi that, that are able to act on these sugars and break them down. So finally, in summary, I just want to point out again that we've been able to show active expression of many of these CAZymes in the AMD environment. And secreted enzymes from A. richmondensis have adapted to these extreme conditions of high temperature and low pH and high metals. And therefore, they may prove useful for the deconstruction of biomass because of their activity in these conditions. Future work includes characterizing some of these enzymes and also doing a more comparative genomic approach to compare it with other fungi. As I mentioned in the beginning, this work was part of a broader group of people. Jill Banfield and Steve Singer are leading the projects. The sequencing was done at JGI for the metagenome and the genome, as well as the transcriptome work. And we've had a lot of help from a lot of people at JGI on that part. The proteomics was done at Oak Ridge National Lab, and we received additional funding by the DOE carbon cycling program. Finally, I'd like to thank you guys and take any questions that you might have.
Annika, maybe time for one quick question.
No.
So you mentioned that, or you found some alpha-linked glucan or alpha amylases. Mm-hmm. So what do you think they're doing there? Because it seems like amylose wouldn't be a polysaccharide that's likely to be found in the acid mine drainage community.
Are you asking who's making it, or
yeah,
why
I
are
mean,
they
have you
using
seen
it?
any amylose?
Yeah, we have not seen any amylose, and we haven't actually gone in and looked at who's making it at this point. It's certainly an area that we're looking at because we see that again and again showing up as one of the most highly expressed GHs. Thanks.
Okay, thank you.